Good morning, student. Today we are going to discuss one practical aspect, that is bacterial transformation. And the first step in bacterial transformation is the preparation of competent cells. The objective is to familiarize with how cells are made competent, which is the primary step for transformation. Since DNA is a very hydrophilic molecule, it won't normally pass through a bacterial cell's membrane. In order to make bacteria take in the plasmid, vector or any foreign DNA, they must be made competent to take up DNA. This is done by creating small holes in bacterial cells by suspending them in a solution with a high concentration of calcium. DNA can then be forced into the cells by incubating the cells and the DNA together on ice, placing them briefly at 42 degrees, that we call heat shock, and then putting them back on ice. This causes the bacteria to take in DNA. The cells are then plated on antibiotic containing media. The diagram here showing the normal bacterial cell, if you see the right hand side corner, and if you treat with ice cold calcium chloride solution, what we are expecting that there will be a small hole on the bacterial cell membrane, and that gap actually, with the help of that pore, the plasmid, which is a foreign DNA, which will intrude inside the cell. And not only that is the reason, if you need to give a heat shock, the shock at 42 degrees centigrade for 1.5 minutes to 2 minutes will open that hole so that the foreign DNA, the vector, can go inside the cell. That type of cell, now you need to maintain the cell in plate them on an antibiotic containing media. The antibiotic resistance property is coming from the vector, the plasmid, which has taken from the gene which it is carrying. The next thing, the principle, the concept, the competent cells are ready to use bacteria cells that possess more easily altered cell walls by which foreign DNA can be passed through easily. The standard method for making the bacteria permeable to DNA involves treatment with ice cold calcium ions. However, some types of bacteria are naturally transformable, which means they can take up DNA from their environment without requiring special treatment. The exact mechanism involved in artificial competence are not yet known. What we understand, that calcium chloride method, it's because of it creates some pores in bacterial cells by suspending them in a solution, high concentration of calcium. And if you give a heat shock, particularly at 42 degrees centigrade, because more than that maybe they are not, cells are not will survive, so that will help the foreign DNA molecule to enter inside the cell. That process we call transformation. So competence is distinguished into natural competence. Some cells have a transformation efficiency is more, that means they are easily genetically specified ability of bacteria that is thought to occur under natural condition. Artificial competence arising when cells in laboratory culture are treated to make them transiently permeable to DNA. So let us see how in the lab we are going to use these competent cells. So here what we are going to take, we have taken bacterial cells, the bacterial cells which supplied in the kit, that is DH5 alpha. You must be knowing this type of bacteria, they are ready to accept any foreign DNA. So plasmid DNA they supplied, this is PAC18 plasmid. We need to prepare 0.1 molar calcium chloride. Ampicillin is already provided. We have ex-gal, we prepared LB broth, agar. So here what we are doing, they are preparing a very healthy bacterial culture in LB broth, and which they are actually it's a maxi prep, that means a large number of culture they have prepared, and they have centrifuged the culture. You can see the pellets are present in this tube. So they are discarding, after centrifugation, what they are doing, they are discarding the media. So with this, the cell pellets are visible. Now they are going to treat with ice cold calcium chloride, 0.1 molar calcium chloride solution. So you can see that it is suspended with ice cold calcium chloride solution, both the tubes, and keeping in the ice 4 degree, and then suspending with a dropper. They are now mixing the cells so that there should not be any clump. So suspend the whole pellet in calcium chloride solution. Again, centrifugation they are doing at 4000 rpm for 15 minutes. Once the centrifugation is done, then again they are taking they're discarding the soup, taking the pellet. They will repeat the whole process 2 to 3 times. So you can see they are discarding the calcium chloride, again adding the calcium chloride solution. So 2 to 3 times centrifugation in a ice cold condition is very much important here. Again need to suspend the cell with the help of a dropper, and then again going for a centrifugation. The same thing repeating, suspending the pellet, and then after centrifugation, after suspending with calcium chloride solution, what now they are doing, 100 microliter of this suspension meaning calcium chloride and the pellet, they are aliquoting in each tube approximately 100 microliter, and these are the cells, competent cells are ready for transformation. That means heat shock, which is the second step. You can use the competent cells immediately, or you can store it in liquid nitrogen or minus 70 degree any refrigerating condition. Thank you very much.